Have you ever wondered how something as intangible as an idea, a gene sequence, or a living organism can actually be owned under the law? In today's session, we'll understand protectable subject matters, protection in biotechnology, protection of other biological material, and ownership and period of protection, covering everything from copyright and trademarks to patents and gene patenting. By the end of this lecture, you'll clearly understand not just the definitions, but also the exact time periods and ownership rules that are frequently tested in exams. Let's begin by understanding intellectual property. IP refers to the creations of human mind, like inventions, literary and artistic works, and symbols, names, images, and designs in commerce. IPRs protect the interests of creators by giving them property rights over their creations. A Venetian law of 1474 made the first systematic attempt to protect inventions by a form of patent, which granted an exclusive right to an individual for the first time. In the same century, the invention of movable type and printing press by Johannes Gutenberg around 1450 contributed to the origin of the first copyright system in the world. Importance and the role of the IP protection has been crystallized in the Trade-Related Intellectual Property (TRIPS) agreement. It establishes standards for availability, scope, use, enforcement, acquisition, and maintenance of IPR. It addresses related dispute prevention and settlement mechanisms. Remember this because it is frequently asked in examinations, especially the 1474 Venetian law and the 1450 Gutenberg printing press, since these are the earliest historical roots of IP protection. Moving on to subject matter protected by IPRs. The first point is Paris Convention for the Protection of Industrial Property, 1883, and the Berne Convention for the Protection of Literary and Artistic Works in 1886. The next point is Convention Establishing the World Intellectual Property Organization, 1967. And finally, protection against unfair competition and all other rights resulting from intellectual activity in the industrial, scientific, literary, or artistic fields. If you observe the illustration, this slide gives a visual overview connecting the different categories of intellectual property that we're about to study one by one, starting with copyright, moving through trademark, geographical indications, patents, and finally, industrial designs. Keep this point in mind because we'll be going through each of these categories in detail across the next several slides. Let's continue with copyright. Copyright is a right given by the law to the creators of literate, dramatic, musical, and artistic works and produce cinematograph films and sound recordings. Creator of copyrighted work has the right to control or prevent unauthorized copying or reproduction of the work by others for a certain time period. The protection of copyright varies according to national legislations and type of work. The Indian law extended copyright protection for the work made by an individual for lifetime of the author plus 60 years. Remember this because it is frequently asked in examinations. The duration is lifetime of the author plus 60 years under Indian law. Building on that idea, let's discuss trademark. Trademark is the most valuable asset owned by businesses. Businesses with particularly successful products or services spend considerable amount of time, effort, and money creating, establishing, and promoting their unique identities. In India, trademark registration is valid for a period of 10 years. The same may be renewed from time to time for additional period of 10 years. Let's look at international protection for trademark. An application has to be filed in each and every country where one wants protection for their trademark, and also has to be made in relevant class of goods or services over which the mark is used. There are two different ways. The first option is to file application individually in each and every country where the applicant wants protection for their trademark. The second option is to file application for trademark protection through regional conventions, which provide for registration in multiple countries by single application process. The next concept we'll discuss is geographical indications. GIs refers to indication which identifies a good as origin in the territory of a member, or a region or locality in territory where a given quality, reputation, or characteristics of the good is essential attributable to its geographical origin. In India, GI may initially be registered for a period of 10 years and can be renewable from time to time for further periods of 10 years. Indian law certain restrictions in that GI registered is not a subject matter of assignment, transmission, licensing, pledge, mortgage, or any such other agreement. Think about this for a moment. GI protection is similar to trademark in duration, but unlike a trademark, it cannot be transferred or licensed to someone else, since it belongs to a whole region rather than an individual owner. Another important topic is patent. A patent is a form of protection that provides a person or legal entity with exclusive rights for making, using, or selling a invention, and excludes others from doing the same, claiming damages from those who infringe the invention. It is granted by Indian Patent Office and has a term of 20 years. After expiration of this 20-year monopoly, the product invention will fall in public domain for any third party to use it. Let's look at ownership of patent. In most countries, both natural persons and corporate entities may apply for a patent. In the United States, however, only the inventor may apply for a patent, although it may be assigned to corporate. In most European countries, ownership of an invention may pass from the inventor to their employer by rule of law. This is one of the most important concepts in this chapter. The 20-year patent term and the difference in ownership rules between the US and Europe are commonly asked comparison questions. Let's shift our attention to industrial designs. ID refers to the creation of a shape, configuration, or composition of pattern or color in 3D forms containing aesthetic value. Designs Act only protects designs that are aesthetic in nature. An ID can be a 2D or 3D pattern. Novelty and originality are important criteria for a design registration. The Industrial Design Registration grants the exclusive rights of selling, importing, and applying the design to any product. In India, a design registration is valid for a period of 10 years, renewable for further periods of 5 years. This brings us to agricultural biotechnology. Encompasses a range of research tools used by scientists to understand and manipulate the genetic makeup of organisms for use in agriculture. Let's continue with major benefits. Under direct benefits, the first point is improving the nutritional quality of foods, for example, golden rice. The next point is reducing the presence of toxic compounds, for example, cassava with less cyanide. Another point is reducing allergens in certain foods, for example, groundnut. Can you relate this to a real-life example? Golden rice is a very commonly cited case in exams for nutritional biofortification through biotechnology. Moving further, let's discuss why IP in biotechnology. R&D in biotechnology is extremely time-consuming, requires huge investment, granting IP an effective way to protect biotechnology inventions. One more important concept is the US-EU dispute. US is the world's largest grower of GM crops. GM crop activity in EU member states is minimal, partly because the EU only ended a 6-year moratorium on growing GM crops in 2003. Individual applications to import GM seeds into the EU need the approval of all 25 member states. Some EU member states, such as Australia, Germany, and Italy, remain strongly opposed to growing GM crops. Europe's approach is based on the precaution principle. Though commercial GM crops are banned, food in the EU is allowed to contain GM ingredients, but any food product whose GM content exceeds a certain percent needs to be labeled. This is because of another principle underlying EU policy, that the general public should be able to choose whether to consume GM food or not. Many large European supermarkets have chosen to remove GM ingredients from their products. Campaigners in Europe want even stronger legislation to ensure that GM produce should be kept separate from non-GM produce at every stage of production. Notice how this connects with the previous topic on agricultural biotechnology. This slide shows us the real-world tension between promoting biotech innovation and addressing public concern. Let's continue with IPR in biotechnology. IPR for biotechnology is currently in a state of flux. It used to be the case that living organisms were excluded from protection, and increasingly, biotechnology is receiving some form of protection. These changes have largely taken place in the USA and other industrialized countries, but as other countries wish to compete in order to protect and encourage investment in biotechnology. Biotechnological inventions fall into three categories. The first category is processes for the creation or modification of living organisms. The next category is biological material, the results of such processes. And the third category is the use of such results. Remember this because it is frequently asked in examinations. These three categories of biotechnological inventions come up often as a direct list-based question. Another important topic is IPR currently available. There are two main systems of protection for biotechnology: rights in plant varieties and patents. Keeping biotechnology as a trade secret can also be a valuable form of protection, which will provide protection against those who steal other trade secrets unfairly. However, the problem with this form of protection is that the secret generally becomes public once the biotechnology is used commercially, and thus the protection is lost. When it comes to protection with copyright, it is generally unsuitable because of practical and conceptual difficulties. Let's look at examples of patents in biotechnology. 1980, first Cohen-Boyer recombinant DNA patent. 1984, cell line derived from human leukemia patient patented. 1986, genetically engineered corn patent, BT corn. 1988, Harvard oncomouse patent. 1991, isolated human bone marrow stem cells patent. This is one of the most important concepts in this chapter. This timeline of biotechnology patents from 1980 to 1991 is a favorite exam question, so try to remember at least the years and the corresponding inventions. Let's shift our attention to international treaties related to biotechnology. There are three international intellectual property treaties which are of particular importance for the protection of biotechnology. The first is Paris Convention for the Protection of Industrial Property, the Paris Convention. The next is the Budapest Treaty on the International Recognition of the Deposit of Microorganisms for the Purposes of Patent Procedure, the Deposit Treaty. And the third is the Patent Cooperation Treaty, PCT. Moving on, let's discuss deposit of biological materials. In India, this is done at IMTech, Chandigarh. Let's look at what to deposit: GMOs, bacteria, viruses, cells, cell line, seeds, plasmids. Now, why deposit? Viability, 30 years. Keep this point in mind because we'll use it again later. IMTech Chandigarh and the 30-year viability figure are commonly asked one-mark questions. This brings us to gene patents. What exactly is patentable? If you observe the table, it lists different types of genetic material against whether they are patentable. Raw DNA sequence? The answer is no. Full-length genes, cDNAs with function? The answer is yes. Partial sequences, ESTs and SNPs? The answer is no. With computer-aided guess of function? The answer is sometimes. With biological info showing function? The answer is yes. Entire genomes? The answer is no. This is one of the most important concepts in this chapter. Students often get confused between these categories, so remember, function-proven full-length genes are patentable, but raw sequences, partial sequences and entire genomes are not. Let's continue with ownership of patent. In most countries, both natural persons and corporate entities apply for a patent. In the United States, however, only the inventor may apply for a patent, although it may be assigned to a corporate. In most European countries, ownership of an invention may pass from the inventor to their employer by rule of law. Owner of copyright in a protected work may use the work as he wishes and may prevent others from using it without his authorization. The rights granted under national laws to the owner of copyright in a protected work are normally exclusive rights to authorize a third party to use the work, subject to the legally recognized rights and interests of others. Finally, let's discuss expiry of a patent. A patent can expire in the following ways. The first way is the patent has lived its full term. The next way is the patent has failed to pay the renewal fee. Another way is the validity of the patent has been successfully challenged by an opponent by filing an opposition with the patent office or with the courts. As soon as the patent expires, it passes to the general public domain, and now anybody can use it without the permission of the original inventor. Remember this because it is frequently asked in examinations. These 3 ways a patent can expire are a common short answer question. Thank you. And that concludes today's teaching slides. If you found this lecture helpful, please like the video, subscribe to our channel, and share it with your friends. If you have any questions, feel free to leave them in the comments section. Thank you for watching, and I'll see you in the next lecture.